Hello, my name is Diego. I'm an undergraduate student studying Earth Systems on the Sustainable Food and Agriculture track. Our lab incubation had three main questions we were looking at. The first one was what is the effect of temperature on cover crop breakdown? And so we had three different temperature schemes which reflected the average temperatures during the winter and autumn growing seasons in the Salinas Valley. We also were wondering about the effect of carbon to nitrogen ratio on cover crop breakdown, and so we had various carbon to nitrogen ratios ranging from 10 to 1 up to 30 to 1 to understand how much nitrogen was produced relative to that starting carbon to nitrogen ratio. And finally, we were looking at residual nitrogen, or the amount of nitrogen left in the soil, and understanding the impact that has on breakdown as the incubation progressed.
Hi, my name is Anna Gomes and I am a PhD candidate at Stanford University working on the cover crop team. And today I'm going to tell you about setting up our incubation experiment. We collected over 300 pounds of soil from the top 30 centimeters of our field site. After we collected and prepared the soil for the incubation from the field site, then we got to work figuring out how much cover crop biomass would go in each tube. We calculated an application rate of about 7,000 pounds per acre to fit in each tube, which turned out to 1.6 grams of biomass. Once the tube received its biomass and soil and also its moisture, then it was time to cover the top of the tube with parafilm to retain the moisture and also take the final mass so that we can maintain the moisture over the duration of the experiment. The tubes were now ready to be put inside the incubation temperatures. 3 temperatures were included in the experiment: 20 degrees Celsius, 15 degrees Celsius, and 10 degrees Celsius. By measuring the plant available nitrogen in our soil samples in our PVC tubes over the time of 16 weeks, between the different treatments, we can then tell how fast the plant biomass or the cover crops are decomposing and releasing their nitrogen from organic nitrogen to inorganic nitrogen. Our research asked a simple but important question: how do the carbon to nitrogen ratios of cover crop residues and soil temperature interact to determine the timing and magnitude of nitrogen release into the soil? At optimal soil moisture, we found that the carbon to nitrogen ratio was the strongest predictor of nitrogen mineralization. Residues with a C to N ratio like C to N10 release large amounts of plant available nitrogen, as noted here on the plot as %N-min or %N mineralization, over the 112-day study period, while residues with a high C to N ratio like 30 to 1 often immobilize nitrogen, meaning microbes tied up soil nitrogen instead of releasing it. Soil temperature added another layer. Warmer conditions accelerated decomposition, but this effect was most pronounced for the residues with a high C to N ratio. For example, at 20 degrees Celsius, even the residue with a C to N of 30 eventually released nitrogen. We also investigated whether adding extra inorganic nitrogen could boost mineralization of high C to N residues. Surprisingly, it made little difference at the rate that we applied. Altogether, our results highlight a trade-off. Low carbon to nitrogen ratio cover crops provide nitrogen quickly, but risk nitrogen losses via leaching if terminated in the fall. And high C to N cover crop ratios release nitrogen more slowly, reducing the risk of leaching but potentially immobilizing nitrogen needed for the next season crop. These findings are directly relevant to nitrate leaching policies in California's Central Coast and similar regions. They suggest that careful management of the timing of cover crop termination, combined with residue quality, or the C to N ratio, is key to balancing nitrogen supply with environmental protection. Thank you for watching, and we invite you to read our full article in the Journal of Environmental Quality to learn more.